Welcome to the deep dive. Today, we're embarking on a really fascinating journey into one of the most complex and persistent health challenges we face globally, gastric cancer.
Yeah, it's a big one.
It really is. I mean, despite all our medical advancements, it's still the fifth most commonly diagnosed cancer worldwide. And maybe even more sobering, it ranks third in mortality.
That's right. Those numbers are pretty stark.
They really are. It's a powerful reminder of just how formidable this disease remains.
Indeed. And it highlights just how critical it is for us to, really understand its fundamental underpinnings,
Yeah.
especially at the genetic level. That's where we might find the keys to really turn the tide.
And that's precisely our mission for you today. We've waded through, honestly, a colossal stack of the latest research. We're talking massive genome-wide association studies, GWAS, you'll hear us say that a lot, and also cutting-edge Mendelian randomization analyses.
Lots of data.
Exactly. And we've tried to distill the most pivotal findings. Our goal is really to equip you with a crystal clear understanding of what's really driving this disease from a genetic perspective, moving beyond just the symptoms to the kind of blueprint of susceptibility itself.
So we'll be your guides, hopefully,
Yeah.
through the key genetic hotspots that keep popping up. We'll look at how these big studies are actually designed, which is pretty fascinating in itself.
Right.
And then we'll get into the specific genes and biological pathways that seem to be involved. And some of the really exciting work in what's called fine mapping and functional studies, basically figuring out what these genetic variants actually do.
And we're not stopping there. We're going to dive into some truly transformative developments like polygenic risk scores.
Ah, yes, PRS. Very important.
Yeah, these are powerful tools that are, well, they're revolutionizing how we predict individual risk. We'll also compare findings across different populations, East Asians, Europeans, and others, and explore how our understanding of gastric cancer genetics has dramatically evolved over really just the last decade or so.
It's changed a lot, quite quickly.
Definitely. So think of this deep dive as your shortcut to being incredibly well-informed on the cutting edge of this field. We've got some surprising facts, some practical insights you can genuinely use. So let's unpack this. The genetic landscape of gastric cancer, key hotspots and population differences. All right, let's kick things off by mapping out the genetic landscape of gastric cancer itself. Now, when we talk about uncovering genetic links to complex diseases like this, genome-wide association studies, or GWAS, are often where the real detective work begins.
That's the starting point, yeah.
For anyone tuning in who might be new to this term, GWAS, can you just quickly explain what it is and why it's such a powerful tool in modern genetics?
Absolutely. Think of GWAS as a massive, super high-resolution scan of your entire genetic construction manual, your genome.
Okay.
Researchers compare the DNA of thousands, sometimes hundreds of thousands, of people who have a disease like gastric cancer with thousands of healthy individuals.
Comparing the differences.
Exactly. What they're searching for are common genetic variations, often just a single letter change in the DNA code, we call it SMP, a single nucleotide polymorphism that appears significantly more often in people with the disease.
So you're looking for correlations.
Right. The real power lies in its ability to broadly survey the entire genome without needing a prior hunch about which specific genes might be involved. It effectively shines a light on previously unsuspected genetic regions associated with complex conditions.
That makes perfect sense. It's a bit like searching for specific common typos in a giant book that seem to correlate with a particular outcome, like a health issue.
That's a good analogy, yeah.
So with that incredible power in mind, where are the hot spots? Where have these GWAS studies consistently pointed for gastric cancer? Which regions of our genome seem to be recurrently implicated?
Okay, so one of the most significant and consistently identified regions is on chromosome 5, specifically at a location geneticists call 5p13.1.
5p13.1, okay.
This area involves a gene known as PRKA1. Researchers have found a remarkably strong association here. There's one particular genetic variant, RS1007491, that shows a really robust link to both cardia and non-cardia gastric cancers.
Ah, okay. And just remind us, cardia versus non-cardia.
Right. Cardia cancer affects the upper part of the stomach near the esophagus.
Yeah.
Non-cardia affects the main body and the lower parts.
Got it.
And the statistical evidence linking this PRKA1 region to both is incredibly strong. The p-values are tiny, like 10 to the minus 12 for cardia and 10 to the minus 23 for non-cardia. And the odds ratio suggests it's protective. Actually having the variant lowers your risk. It really points to PRKA1 as a key player in susceptibility.
Wow, that's a powerful discovery, especially linking it to both types, even if it's protective. What about other chromosomal regions that have consistently shown up?
Another important area lies on chromosome 6 at 6p21.1. This is very close to a gene called UNC5CL. Yeah. A specific genetic variant here, RS2294693, has been strongly associated with non-cardiac gastric cancer risk. Interestingly, it only shows a sort of nominal connection to cardiac cancer.
Okay.
So its primary link seems to be with non-cardia, suggesting, again, a genetic distinction in susceptibility based on where the tumor starts in the stomach.
And I remember MUC1 from earlier discussions, maybe in other cancer contexts. Isn't that gene involved quite broadly? How does it play into gastric cancer?
They're absolutely right to recognize MUC1, or mucin-1. It's a gene that encodes a mucin protein. Normally, this protein acts as a protective barrier for our gastric epithelial cells, the stomach lining.
So it's a good guy, usually.
Usually, yes. But what's fascinating and maybe a bit of a biological paradox is that while it's protective in healthy cells, certain variants or dysregulation of MUC1 can be strongly linked to gastric cancer. The specific variant in MUC1, RS407-2037, has been consistently confirmed in gastric cancer studies.
And does that one distinguish between subtypes?
This one actually shows similar risk estimates for both cardia and non-cardia subtypes. It's a great example of a gene where maybe altered function contributes to disease rather than just its presence or absence.
So it's almost like a switch flips in its function, turning something protective into something that promotes cancer.
Something like that, potentially, yeah.
Are there any other familiar names that have appeared? Or perhaps some exciting newer discoveries emerging from these large studies?
Indeed. PSCA, which stands for prostate stem cell antigen, despite the name, it's relevant here, was previously pretty well established as linked to gastric non-cardia cancer.
Okay.
However, recent larger studies have, interestingly, not found a strong association with cardiac cancer. So that reinforces this idea again that these anatomical subtypes have distinct genetic underpinnings.
Right, the separation keeps appearing.
Exactly. Now, as for truly novel discoveries, a long non-coding RNA, known as LINC 000240, has been identified.
Okay, long non-coding RNA. That sounds a bit technical.
It does, but think of it simply as a piece of genetic instruction that doesn't actually make a protein itself, but it still plays a crucial regulatory role. It controls other genes.
Ah, look at like a manager.
Kind of. And the ALNC 00240 has been identified as a novel oncogene, meaning it actually promotes cancer growth. Its higher expression in gastric cancer samples is unfortunately linked to worse patient survival.
Wow. And do we know how it works?
Yes, functionally. It seems to act by interacting with and stabilizing another cancer-promoting protein called DDX21. This demonstrates its direct role in sort of driving tumor progression.
That's a profound finding. It really shows how even these non-coding parts of our genome can be critical players. It expands our view beyond just the protein-coding genes.
Absolutely. There's a whole other layer of regulation.
What about even newer variants? Are things still popping up in the latest research?
Oh, the discoveries definitely keep coming. For instance, a variant called RS836545 on chromosome 7 is linked to a higher expression of the DAGLB gene in people carrying the risk allele.
Okay, DAGLB.
Similarly, another one, RS1892252 on chromosome 6, is associated with higher expression of BTN3A2.
BTN3A2.
Okay. Now, what's truly significant here is that DAGLB and another newly identified gene, FBXO43, are both found to be significantly overexpressed in actual gastric cancer tissues. And unfortunately, this overexpression is associated with a less favorable prognosis for patients.
Not good news for those genes to be highly active, then.
No. And when researchers looked at the networks these genes operate in the pathways they're part of, they found that their co-expression genes, the ones they work with, are enriched in crucial, well-known cancer-related pathways. Things like P53, MAPK, and TGF-beta.
Ah, the usual suspects in cancer biology.
Oh, exactly. So this isn't just a random list of gene names. It tells us these newly found genes are directly plugging into the fundamental cellular machinery that drives cancer. They're not isolated players.
So they're not isolated anomalies. They're integral to these core cancer pathways. That paints a much clearer picture of the biological mechanisms potentially at play. Any other exciting gene discoveries or mechanisms that have caught researchers' attention recently?
Absolutely. There's a gene called C1-GESTRI-LT1. A specific variant in this gene, RS359995883, seems to play a critical role in gastric cancer development. It was associated with risk.
Yeah.
Again, looking like a protective effect, actually.
Interesting. And functionally?
What's intriguing is that higher C1lt1 mRNA levels, meaning the gene is more active, are linked to worse overall survival. And it correlates positively with immune cells, like CD4 plus T cells and macrophages infiltrating the tumor.
Ah, so it might be involved in shaping the tumor's immune environment.
That's the suggestion, yes, which is a really hot area in cancer research right now.
Definitely. And you mentioned earlier that it's not just the DNA sequence. Epigenetics plays a role.
Yes,
Yeah.
exactly. Beyond direct genetic sequence changes, an area that's showing increasing importance is epigenetic modifications, specifically DNA methylation.
Right, the tags on the DNA.
Think of DNA methylation as chemical tags or sticky notes on our DNA. They don't change the genetic code itself, but they can powerfully influence whether a gene is turned on or off.
Like dimmers or switches.
Precisely. Researchers have found pronounced hypermethylation, basically too many off tags in key promoter regions of genes in gastric cancer tissue compared to normal stomach tissue.
And any specific genes stand out?
The PRKCB gene really stands out here. It shows the most significant disparity in these methylation tags between cancer and normal tissue. This hypermethylation effectively silences PRKCB. It reduces its levels in cancer tissues.
And why is that important?
Well, it highlights its role and potentially conferring risk when it's silenced. And importantly, it opens up new diagnostic possibilities. Maybe we can detect this methylation pattern and even therapeutic perspectives, like finding ways to reactivate the gene.
So it's not just the static blueprint of our DNA, but how it's dynamically read and regulated that truly matters. It's like the software running the hardware.
That's a great way to put it.
And what about genes that maybe they don't hit that super strict statistical significance cutoff in every single massive GWAS, but there's still strong epidemiological evidence suggesting they're associated with gastric cancer risk? Is there a sort of second tier of suspects?
Yes, absolutely. Beyond these top-tier findings that cross the genome-wide significance threshold, a number of SNPs and other genes have presented strong, cumulative epidemiological evidence for an association with gastric cancer risk, maybe nominally significant in multiple studies.
Okay.
These include variants in genes involved in DNA repair like APE1 and ERCC5, methylation like DNMT1, detoxification like GSTT1, cell cycle control like MDM2, metabolism like PPRG, inflammation like TLR4 and IL-17F, and apoptosis like CSP8.
Wow, that's quite a list. It covers a lot of biological ground.
It really does. This broader list underscores the complex, multi-gene nature of gastric cancer susceptibility. It suggests that many different subtle genetic variations likely contribute to an individual's overall risk profile. It's not just one or two genes.
That's a huge list of candidates. Now, you mentioned anatomical specificity earlier, like cardia versus non-cardia cancer. Does this genetic heterogeneity extend to the different histological subtypes of gastric cancer? Meaning how the cancer cells actually look under a microscope, like intestinal versus diffuse types, are they genetically distinct too?
It absolutely does, yes. GWAS analyses strongly underscore that these gastric cancer subtypes indeed display distinct genetic architectures. For instance, a very large GWAS, the one involving over 5,800 patients and nearly 11,000 controls, specifically highlighted these subtype-specific associations.
So it matters not just where the cancer is, but what it looks like microscopically.
Exactly. This genetic heterogeneity is a critical nuance. It moves us away from a potentially simplistic, one-size-fits-all understanding of gastric cancer towards a much more precise, personalized view based on the specific tumor characteristics.
Can you give us some examples of how these subtypes differ genetically, or how specific genes might be more relevant to one type than another?
Well, it's some of the risk loci we've discussed. Increased expression of genes like MUC1, ANKRD50, PTGR4, and PSCA have been identified as plausible mechanisms driving gastric cancer. And their specific level of upregulation might differ between, say, intestinal and diffuse subtypes.
Okay.
What's also quite fascinating, and connects back to older observations, is the influence of blood groups. Genetics is now helping us understand this better.
Ah, yes, the ABO blood groups.
That's right. Blood group O was found to offer some protective effects, specifically against non-cardia and diffuse gastric cancer, while blood group A, interestingly, seemed to increase the risk for both subtypes.
That's a truly surprising fact about blood groups. You wouldn't immediately connect that to cancer risk, but the link seems real.
It's a classic epidemiological observation that genetics is now helping us potentially understand the mechanism behind.
Are there any other subtype-specific associations that stand out as particularly important for how we might treat or manage the disease down the line?
Yes, there's a specific genetic variation in a gene called C12R51, known as RS11066280. This variant was uniquely associated with poorer survival outcomes, but specifically for intestinal-type gastric cancer.
Only intestinal type.
That's what this study found, yes.
Mm.
And this detrimental effect was even more pronounced in patients with larger tumors, over 5 centimeters, those without distant metastasis, strangely enough, and also notably in patients undergoing chemotherapy or who were drinkers.
Wow, that's quite specific.
It is, and this discovery highlights its potential utility as a prognostic marker. It could potentially help guide treatment decisions based on an individual's genetic profile, combined with their tumor characteristics and lifestyle factors. It's a really powerful step towards personalized medicine.
So genetics aren't just about identifying who's at risk initially, but also potentially predicting how a patient might respond to treatment or how their disease might progress. That's incredibly valuable information.
Exactly, it's about tailoring the approach.
Now, let's talk about populations. Gastric cancer incidence varies wildly around the globe, right? With East Asia having particularly high rates.
Tragically high, yes.
So how do GWA's findings compare across different populations, especially East Asians versus Europeans? Do we see the same genetic culprits popping up everywhere, or are there distinct regional differences?
That's a crucial point. And you're right, a significant proportion of the GDA's findings, especially some of the most impactful ones, actually originate from studies conducted in East Asian populations.
Why is that? Just the higher incidence?
That's a major reason. It naturally reflects the historically high incidence of gastric cancer in regions like Japan, Korea, and China. This means researchers there have access to large patient cohorts, which are essential for conducting statistically robust studies to discover these associated genetic variants.
Makes sense. Can you give us some concrete examples from, say, Korean studies, given their significant contribution to this field?
Certainly. A large case control study in Koreans involving over 3,200 gastric cancer cases and 1,700 controls found that a particular genetic variation, the RS13361707 polymorphism, located within those PRKAA1 and PTGR4 genes we mentioned.
Ah, back to PRKAA1.
Yes, this variant was significantly associated with increased gastric cancer risk in this Korean population. Carriers of specific genotypes, TC or CC, showed higher odds of developing the disease compared to the TT genotype. This adds more weight to the role of these genes.
Okay. Any other examples from Korea?
Well, beyond direct cancer associations, other Korean GWOs have identified SMPs, like those in the NRG3 gene, as highly associated with metabolic traits like basal metabolic rate and BMI, specifically in obese Korean women.
How does that relate to gastric cancer?
It's an indirect link, potentially. But given that obesity is a known risk factor for several cancers, including potentially gastric cancer, understanding the genes involved in metabolism and body weight regulation in specific populations might offer new insights into cancer risk pathways eventually. Some of the genes involved, like BCL2L2, PABPN1, or SLC22A17, might be interesting to look at in the cancer context too.
That's interesting, sort of connecting the dots between metabolism and cancer risk genes. But what about the elephant in the room when it comes to gastric cancer, especially in East Asia? The bacterium Helicobacter pylori. How do genetic studies specifically account for or interact with that powerful environmental cofactor?
That's a critical stratification and a brilliant example of gene-environment interaction. Some researchers have been very smart about this. A Korean GWA specifically analyzed its data by stratifying participants based on their H. pylori infection status, positive or negative.
Ah, so they split the groups.
Exactly. This innovative approach revealed that a specific SNP, RS2671655, on chromosome 17q21.33, was significantly associated with gastric cancer risk only in individuals who were H. pylori positive.
Wow, only in the infected group.
Yes. The risk allele, T, was much more common in cases who had the infection. This variant showed no association in the H. pylori negative group. This powerfully highlights how a particular genetic variant might only confer risk, or perhaps significantly increase risk, when combined with the presence of this specific bacterial infection. It's a perfect illustration of how our genes and our environment can, well, conspire.
That really underscores the interaction. And speaking of H. pylori, I read that Mongolia has the world's highest gastric cancer prevalence. Is that right?
It is, tragically, extremely high rates.
Are there specific insights from studying the H. pylori genomics in that region that might shed light on this extreme incidence? Is it the bacteria itself?
It seems to be a major factor, yes. In Mongolia, research found that nearly all H. pylori strains isolated from gastric cancer cases, an astounding 93.8%, belong to a highly virulent subgroup known as SG1.
Highly virulent. Okay.
What's particularly striking is that even though these strains often have a Western-type CAG-A protein, which is one virulence factor,
Mm-hmm.
they almost universally possess a complete and highly prevalent CAG-PAI.
The CAG-PAI.
It's a crucial pathogenicity island, a chunk of genes that carries many key virulence factors. The hypothesis is that the completeness and high prevalence of this CAG-PII in Mongolian strains, coupled perhaps with specific host genetic factors and other determinants within the H. pylori genome itself, are likely driving Mongolia's exceptionally high gastric cancer rates. It's probably about the potent combination of bacterial virulence factors and host genetics, rather than just the simple presence or absence of one specific factor like CAG alone.
So it's not just if you have H. pylori, but which specific strain you have, how virulent it is, and how that particular strain interacts with your own unique genetic background. That adds layers and layers of complexity.
It really does. It's a complex interplay.
How do these findings, particularly those from East Asia, where H. pylori is so intertwined, compare when we look at European populations? Do we see shared genetic susceptibilities, or are there distinct differences that really stand out?
Well, there are both shared commonalities and some quite distinct differences.
Mm.
For instance, take a variant in the IL-17A gene, RS2275913. IL-17A is involved in inflammation.
Okay.
A large meta-analysis, pooling data from 21 studies with thousands of patients and controls, found this variant was consistently associated with increased gastric cancer susceptibility among Asian subjects. Carriers of the A variant were about 1.2 to 1.4 times more likely to develop GC.
That in Caucasians.
For Caucasian subjects, however, the same association was observed, but primarily in a recessive model, meaning you likely needed 2 copies of the risk allele to see the effect. This highlights, potentially, population-specific genetic effects. The same gene might influence risk differently, depending on the genetic background or maybe environmental interactions common in that population.
So the same gene, but potentially different risk models or mechanisms in different populations. That's a key distinction to make.
Absolutely. It cautions against generalizing findings too readily across ancestries.
What about something like serum uric acid? We hear about gout, but does it relate to gastric cancer? And does that show any population differences?
That's an interesting one. They looked at using Mendelian randomization. They compared large European and East Asian cohorts, hundreds of thousands of people,
Yeah.
and they found no significant causal linkage between genetically predicted serum uric acid levels and gastric cancer risk in either population.
So probably not a major driver then?
It suggests that this particular factor, despite maybe showing up in some observational studies, likely does not widely influence risk across these diverse ancestries in a causal way. It helps clarify what truly drives the disease versus what might just be an incidental correlation or confounding. MR is great for that. 2. Methodological approaches, from GWAS to functional insights and risk prediction.
Okay, that's a really comprehensive look at the genetic hotspots, the subtypes, the population differences. It's a complex map. Now, let's shift our focus a bit and talk about how this incredible science is actually done. The sheer scale and complexity of these genetic studies are, frankly, mind-boggling.
They really are massive undertakings.
Can you walk us through how study designs have evolved, the kinds of sample sizes involved, and how researchers ensure that the findings they uncover are truly robust, especially when you're dealing with so many potential confounding factors like lifestyle or environment?
Yeah, the field has evolved tremendously, definitely utilizing increasingly diverse and, crucially, massive cohorts. To give you a sense of scale, some of the largest GWAS, particularly those looking at general gastric cancer subtype associations, have included cohorts as vast as, say, nearly 6,000 patients and almost 11,000 controls.
Wow, thousands and thousands.
Exactly. This provides substantial statistical power to detect common genetic variants, even those that might have relatively small individual effects on risk. Other studies, like that Korean GWAS we discussed looking at specific polymorphisms, involved, over 3,000 cases and 1,700 controls. And studies focused specifically on linking genetic variants directly to gene expression. We call these ESMP or EQTL studies, have leveraged data from thousands more individuals as well.
And when we talk about Mendelian randomization, which seems to be the real cutting edge for trying to infer causality,
Yeah,
those numbers get even bigger, right?
precisely. Mendelian randomization, or MR studies, which, remember, are designed to infer a causal relationship by using genetic variants as natural, unconfounded proxies for exposures like obesity or cholesterol levels.
Right, using genes as a sort of natural experiment.
Exactly. These MR studies often utilize truly colossal data sets. For instance, one study looking at the potential causal role of the cholesterol drug simvastatin on gastric cancer encompassed a staggering 460,000 participants for the simvastatin exposure genetics and over 470,000 for the gastric cancer outcome genetics, all of European descent.
Good grief, nearly 500,000 people in each arm.
Yes. These massive sample sizes are absolutely crucial for identifying potentially subtle causal relationships and, importantly, overcoming the inherent limitations of traditional observational studies, like confounding, where lifestyle factors mix things up, and reverse causality, where the disease might affect the exposure rather than the other way around.
That's an incredible amount of data to process. How do they actually collect all that genetic information? What kind of lab techniques or genotyping platforms are commonly used? And how do they ensure they get comprehensive coverage of the whole genome?
Right. Well, researchers employ a variety of sophisticated genotyping methods. It depends on the scale and the specific questions of the study. These include established techniques like PCR-RFLP, PCR-SSCP for smaller scale studies, and then more high-throughput platforms like Sequenom-SRA, TOCMan assays, and particularly powerful SNP arrays, sometimes called SNP chips, like the 48-plex SNP scan, or much larger arrays from companies like Illumina or Affymetrix.
So chips that can read hundreds of thousands or millions of SNPs at once.
Exactly. But even then, you don't genotype every single SNP in the genome. So beyond directly measuring SNPs, a common and incredibly powerful technique is imputation.
Imputation, filling in the gaps.
That's precisely it. This involves statistically inferring the genotypes of unmeasured SNPs by using large, well-characterized reference panels, like the HapMap3 project or the 1000 Genomes Project data, or even larger ones now. These panels contain genome sequences from many individuals.
So you use known patterns of how SNPs are inherited together.
Exactly. You use the patterns in the reference panel to predict the SNPs you didn't directly measure in your study participants, based on the SNPs you did measure. This effectively fills in the blanks in the genetic data, significantly increasing the genomic coverage and the statistical power of the studies, without having to directly genotype every single one of the millions of common variants.
So they're not just looking at the variants they directly measure, but also inferring millions of others. That really broadens the scope and the depth of the analysis significantly.
It's a standard and essential part of modern GWAS.
Okay. But with something as complex as gastric cancer, where lifestyle and environmental factors, smoking, diet, infections like H. pylori play such a huge role, how do these studies manage to control for all those known risk factors? How do they isolate the genetic effect from all that noise?
That's a critical aspect, absolutely, to ensure the observed genetic associations are truly genetic and not just reflections of, say, smoking habits that happen to correlate with a particular genotype.
Right. You need to untangle it.
You do. And studies are meticulously designed to diligently account for known risk factors. For instance, information on age, gender, smoking status, sometimes alcohol consumption, socioeconomic status, and other relevant factors are collected. These are then routinely included as what we call covariates in the statistical analyses, typically logistic regression models.
So you adjust the math for these factors.
Exactly. This allows researchers to statistically minimize confounding. You're effectively adjusting for these factors to try and isolate the specific genetic effects from that tangled web of potential environmental and lifestyle influences.
And we saw a great example of that earlier with H. pylori. Can you elaborate a bit more on how stratifying by H. pylori infection status is particularly important and methodologically valuable in gastric cancer GWAS, especially in those high prevalence areas?
It's absolutely crucial, especially in regions like Korea or Japan, where H. pylori prevalence is or was historically very high. As we discussed, some GWAS specifically stratify their analyses. They literally split the participants into two groups, H. pylori positive and H. pylori negative, and run the GWAS separately in each group.
Why is that so powerful?
Because it allows researchers to identify genetic variants that specifically modulate risk only in the presence or absence of H. pylori. The RS2671655 SNP we talked about is the prime example. A significant association with gastric cancer risk was found only in H. pylori positive individuals. This kind of meticulous stratification is vital for unraveling the truly complex interplay between inherited genetics and microbial infections and driving carcinogenesis. It's where the environment, in this case the bacteria and our genes, literally shake hands to influence risk.
That's a fantastic transition, actually, because it brings us right to the next big question. Once you identify an association, once you find a SNP that seems linked to the disease, how do you figure out what that genetic variant actually does biologically?
Right. The so what question.
Exactly. This moves us from simply observing a correlation, an association, to understanding the actual biological cause or mechanism. This is often referred to as fine mapping and functional work. So, how do scientists bridge that huge gap between finding a suspicious SNP on a GWAS plot and understanding its precise biological function?
This is where the field truly deepens and honestly becomes incredibly exciting, but also very challenging. Beyond just identifying statistical associations, researchers are now intensely focused on delving into the functional consequences of these genetic regions, these loci, identified by
Trying to
GWAS.
figure out the why.
Precisely. And as we noted, many of these identified SNPs fall into non-coding regions of the genome, the parts that don't directly provide instructions for making proteins.
The so-called junk DNA that isn't junk at all.
Exactly. That term is long outdated. This immediately suggests that their role might be in regulating how other genes are expressed. Are they turning genes up or down?
Okay.
Studies identifying those novel variants like RS836545 and RS1892252 illustrate this beautifully. Remember, individuals carrying the risk allele of these variants exhibit higher expression of the nearby genes, DAGLB and BTN3A2, respectively.
Right, the risk allele cranked up the gene's activity.
Correct. And critically, these genes are then found to be significantly overexpressed in actual gastric cancer tissues and linked to a poorer prognosis. So this directly connects the genetic variation to a functional, potentially disease-driving outcome at the gene expression level.
Yep.
We call these variants ESNPs or EQTL's expression quantitative trait loci.
So it's not necessarily the variant itself causing the problem directly, but rather how that variant changes the way a specific gene is expressed, almost like turning up the volume knob on a particular genetic instruction.
That's often the case for non-coding variants, yes. It's about regulation.
And what about how these genes fit into the larger biological picture? Do they act in isolation, or are they part of broader networks or pathways?
That's the next crucial step. Functional studies reveal that genes identified through GWAS are almost never acting alone. They are typically part of, or enriched in, crucial biological pathways already known to be involved in cancer development or progression.
So they plug into existing systems.
Exactly. For DAGLB and FBXO43, for instance, the genes that tend to be co-expressed with them, the genes they work alongside, are significantly enriched in those well-established cancer pathways like P53 signaling, the MAPK pathway, and TGF-beta signaling.
Right, the big players.
Precisely. This isn't just a list of associated genes anymore. It provides deep insights into the actual biological mechanisms and the cellular machinery driving gastric cancer development. It tells us how these genetic changes might be contributing to the disease at a network level.
That's a big step forward in understanding moving from a list to a mechanism. And you also mentioned epigenetics earlier, specifically DNA methylation, as another way genes can be regulated without altering their sequence. How does that fit into this functional work in understanding the underlying mechanisms of gastric cancer?
This is a fascinating and rapidly growing area, absolutely. Epigenetic modifications, particularly DNA methylation, are key to understanding how gene expression is regulated dynamically, often in response to developmental cues or environmental signals.
Those sticky notes again.
Right, those molecular sticky notes on our DNA that tell a gene to turn on or off without changing the underlying ATCG sequence itself. Researchers have observed pronounced hypermethylation and excess of these off notes in specific promoter regions, the switch regions, of genes within gastric cancer tissues compared to normal stomach tissue.
And you mentioned PRKCB before.
Yes, the PRKCB gene in particular shows the most significant hypermethylation in gastric cancer. This effectively silences PRKCB, meaning its protein product levels are significantly reduced in the cancer cells.
And that silencing contributes to risk.
That's the conclusion. It contributes to gastric cancer risk. And crucially, this finding suggests PRKCB methylation status could be a new diagnostic marker or perhaps even a therapeutic target. Maybe we could find drugs to reverse that silencing?
So we're talking about changes on top of the DNA sequence that profoundly impact its function. And if these are like sticky notes, can environmental factors actually put these notes on our DNA, influencing our risk down the line? Is there an environmental epigenetics angle?
Yes, definitely. Environmental epigenetics is a very active area of research exploring exactly this. There's evidence, for example, that the hypermethylation of a specific DNA site, a CpG site called CG16AuTO7185, might be causally linked to exposure to certain environmental chemicals, specifically PCB99, a type of polychlorinated biphenyl.
Wow, an environmental toxin linking to an epigenetic mark.
Potentially, yes. And this hypermethylation in turn is thought to decrease the expression of the TMX1 gene. Now, TMX1 is normally associated with a better prognosis when it's highly expressed. It seems to play a role in controlling cell proliferation.
So the toxin silences a potentially protective gene.
That's the hypothesis. This illustrates a remarkable potential causal chain from an environmental exposure, PCB99, leading to an epigenetic modification, hypermethylation of CG1-600-71OD5, which then affects gene expression, decreased TMX1, and ultimately influences cancer prognosis. It's a powerful testament to the intricate interplay between our surroundings and our internal biological machinery.
That's a really compelling example of how external factors might influence our internal genetic machinery via epigenetics. And just going back quickly to that new oncogene you mentioned, LINC000240, how does its functional role exemplify what we're learning about these non-coding RNAs?
Right, LINC000240 is a prime example of a non-coding genetic element that we now understand has a direct, active functional role in cancer. Its oncogenic activity isn't just a statistical association anymore. Researchers have painstakingly detailed its molecular mechanism.
They know how it works.
To a degree, yes. It interacts with specific proteins, namely DDX21 and USP10, and seems to stabilize them. This stabilization then directly helps drive gastric cancer progression, promoting uncontrolled cell proliferation, migration, and metastasis. This level of granular mechanistic understanding about how non-coding RNAs exert their influence is a significant leap forward. It's not just association, it's mechanism.
Okay, so we have all this incredibly complex genetic and epigenetic information from single-letter changes in DNA to these chemical tags that regulate gene activity. How are researchers starting to translate all of this complexity into something that might be clinically useful, like actually predicting an individual's risk for developing gastric cancer? Are these polygenic risk scores, or PRS, living up to their hype?
That's the million-dollar question, isn't it? And polygenic risk scores, or PRS, represent a truly powerful tool that aims to do just that. It moves us beyond looking at just one or two high-impact genetic variants.
Right, it aggregates risk.
Exactly. PRS aggregates the effects of many, sometimes hundreds or even thousands, of genetic variants across the entire genome, most of which have only tiny individual effects. It calculates a single score that estimates an individual's overall genetic predisposition to a disease like gastric cancer. It's a way of summing up your inherited genetic burden from countless small contributions.
So essentially, it's a weighted sum of all those tiny genetic contributions, giving you a bigger picture, a more comprehensive estimate of your inherent genetic risk. How predictive are these scores proving to be for gastric cancer specifically?
Well, for gastric cancer, a PRS derived from a substantial set of 112 specific single nucleotide polymorphisms demonstrated remarkably strong predictive power. The association with GC risk was highly significant statistically, with a p-value around 10 to the minus 10.
That sounds pretty strong. How does it translate in practice?
When this PRS was applied to large prospective cohorts, like the China Kadoorie Biobank, CKB, with hundreds of thousands of participants, it showed a very significant stepwise increase in the relative risk of actually developing gastric cancer across different genetic risk groups, or quintiles. For example, compared to those with low genetic risk, the bottom 20%, individuals with intermediate genetic risk had about a 1.5 times higher hazard ratio, meaning they were 1.5 times more likely to get GC over time.
And the high-risk group?
Those with high genetic risk, the top 20%, faced just over a 2 times higher hazard ratio, HR 2.08, compared to the low-risk group. This clearly shows that PRS can effectively stratify individuals based on their inherited genetic risk.
That's already incredibly valuable for identifying high-risk individuals and potentially guiding screening strategies or prevention efforts. But here's where it gets really interesting and, frankly, quite empowering. Does lifestyle still play a meaningful role if you happen to fall into that high genetic risk group? Is your genetic score your destiny?
This is one of the most compelling and, I think, uplifting findings from recent PRS research in gastric cancer and other diseases, too. The answer seems to be a resounding no. Genetics is not necessarily destiny.
That's good to hear.
It is. This PRS study wasn't just about pure genetics. It showed a profound and highly actionable interaction with lifestyle factors. Participants with a high genetic risk score, but who also maintained a favorable lifestyle, things like healthy diet, not smoking, healthy weight, etc.
Yes.
They had a significantly lower risk of developing gastric cancer than those who also had high genetic risk, but lived an unfavorable lifestyle.
How much lower?
The hazard ratio was about 0.53, meaning roughly half the risk. This translated to an absolute risk reduction of about 1.12% over the study period. While that percentage might sound small over a large population, it's substantial. It powerfully underscores that lifestyle choices can indeed mitigate or at least substantially reduce the impact of a high genetic predisposition. It offers tangible hope and clear pathways for prevention, even for those dealt a tougher genetic hand. 3. Unpacking gene-environment interactions and associated factors.
That's an incredibly important takeaway. Our choices genuinely matter, even in the face of significant genetic predisposition. It perfectly sets the stage for our next segment, digging deeper into these complex gene-environment interactions. You mentioned H. pylori quite a bit earlier. How do specific genetic variants truly interact with this common infection, and what other lifestyle factors are coming into sharper focus through this kind of research?
Right. The interaction between genetic predisposition and H. pylori infection is, as we've seen, a recurring and absolutely central theme in gastric cancer research. That SNP we discussed, RS2671655 on chromosome 17.
The one linked only in H. pylori positive people.
Exactly. It was strongly associated with gastric cancer risk, specifically in H. pylori infected Koreans. This isn't a standalone genetic effect. It vividly suggests that certain genetic backgrounds may heighten an individual's susceptibility to H. pylori-driven carcinogenesis. Perhaps it affects the inflammatory response to the bug or the stomach lining's ability to cope with it. It makes them more vulnerable to the bacterial insult and its inflammatory consequences.
So it's not just the presence of the bacteria, but how your body's specific genetic setup reacts to it that's crucial. That's a huge insight, especially if we're thinking about prevention strategies. Is there any evidence then that actually treating the H. pylori infection can make a significant difference, perhaps even more so in those individuals identified with a high genetic risk?
This is indeed a truly compelling finding, and probably one of the most clinically relevant points. Research, including analyses combining PRS with H. pylori status and lifestyle, indicates that H. pylori treatment, especially when combined with maintaining a favorable lifestyle, could significantly reduce the risk of developing gastric cancer, even in individuals identified through PRS as having a high genetic risk.
Wow, so you can potentially counteract high genetic risk with treatment and lifestyle.
Yes, the data suggests that combination is powerful. This translates into a tangible reduction in absolute risk, demonstrating the real-world clinical utility of understanding these complex interactions. It profoundly emphasizes that targeted intervention like eradicating H. pylori and adopting healthy habits based on these combined genetic and environmental insights can potentially have a profound impact on an individual's future health outcomes.
That's a powerful call to action for both public health initiatives, like H. pylori screening and treatment, and for personalized medicine approaches. Now, beyond H. pylori, what about other dietary and behavioral factors that we often discuss in relation to cancer? Are we seeing any specific genetic interactions with, say, what we eat or drink or smoking?
Well, the research consistently touches upon various other environmental and lifestyle factors, although findings specific robust gene diet or gene smoking interactions that replicate widely can be challenging.
Harder to measure consistently, maybe?
That's part of it. For instance, while no conclusive causal evidence was found linking coffee intake genetically to gastric cancer, risk answering a common public question, the consideration of such factors is routine. In those studies on basal metabolic rate and BMI in Koreans, for instance, researchers also observed differences related to family history of stomach cancer and coffee intake among the participants, though not necessarily proving an interaction. More broadly, smoking and drinking status are almost always included as covariates that researchers statistically control for in GWAS analyses.
So they try to remove their influence to see the pure genetic effect.
Exactly. This meticulous approach helps researchers try to isolate true genetic effects from these established environmental risk factors, while still acknowledging their crucial, often larger roles in overall risk. It's often a blend of both genetics and environment lifestyle.
Okay, so scientists are working hard to disentangle the genetic threads from the environmental ones, understanding that it's often a complex mixture driving risk. What about more nuanced internal factors, like specific metabolites circulating in our blood? Are those Mendelian randomization studies shedding new light on those kinds of connections to gastric cancer risk?
Yes, MR studies exploring the human metabolome, the complete set of small molecule chemicals, metabolites found within us, have indeed identified some really intriguing potential connections. It's a newer frontier.
Like what?
For gastric cancer, for example, one study found that the genetically predicted phosphate to alanine ratio was linked to an increased risk. Conversely, the bilirubin to androsterone glucuronide ratio showed an inverse association, suggesting a potential protective effect.
Interesting, specific ratios mattering.
Exactly. And perhaps more intriguingly, 3-methyl-2-oxovalerate, which is an intermediate in branched-chain amino acid metabolism related to protein breakdown, was negatively correlated with gastric cancer risk in an MR study. This hints at potential protective effects, perhaps related to certain metabolic states or possibly even gut microbial activity, as our gut bugs heavily influence our metabolites.
So this could open doors to new biomarkers or even influencing gut bacteria.
That's the hope. These highly specific metabolic findings open exciting avenues for potentially finding novel biomarkers of risk and maybe even developing microbiota-modifying therapeutics in the future, though that's still some way off.
That's incredibly specific. It feels like we're getting a much more granular view of the body's internal chemistry and how it relates to cancer development.
Moving to broader associations now, what other causal inferences are these powerful Mendelian randomization studies giving us? For example, what about obesity? Is there a clear causal link to gastric cancer confirmed by MR, or is it still just a correlation? An MR study provided robust evidence of a causal association between obesity and gastric cancer. The odds ratio was around 1.37, statistically significant.
So obesity causes an increased risk. It's not just linked.
That's what the genetic evidence from MR suggests, yes. It implies that obesity is not just correlated, but acts as a direct risk factor. The mechanism likely involves shared pathways involving chronic inflammation like TNF signaling and cellular growth signals like the PI3K-AKT pathway and general cytokine signaling dysregulation, all of which are known to be affected by obesity and can promote cancer. So yes, addressing obesity could directly impact gastric cancer risk, according to this evidence.
That's a powerful finding, confirming causation. Now, what about other common blood markers, like cholesterol? We often hear about high cholesterol being bad for heart health. Is it the same story for gastric cancer?
This is where things get surprising, actually. Contrary to maybe some common assumptions, a 2-sample MR study found a causal inverse relationship between total cholesterol TC levels and gastric cancer.
Key inverse. So higher cholesterol is protective.
That's what the MR analysis suggested, yes. An odds ratio of about 0.84, significantly below 1. It indicates that higher genetically predicted total cholesterol might actually act as a protective factor, at least in the context of gastric cancer risk.
That's a truly unexpected finding. It really highlights the nuance, doesn't it? What's bad for one disease might not be for another.
Exactly. These complex relationships show that biology isn't always straightforward. What's detrimental for cardiovascular health isn't necessarily so for cancer risk. And sometimes, as seems possible here, the opposite might even be true. Needs more research, but it's a fascinating lead.
Definitely counterintuitive. Now, inflammation is a known driver of many cancers. What have MR analyses revealed about specific inflammatory biomarkers and gastric cancer progression or risk? Are there specific culprits or protectors?
Inflammation is indeed clearly involved and consistently highlighted by Miss All You Know. For instance, MR analysis revealed that genetically predicted levels of the interleukin-6 receptor, IL-6R, which influences IL-6 signaling, a key inflammatory pathway, inversely correlated with gastric cancer progression. So potentially, more IL-6 receptor activity might mitigate risks.
Okay, so that one seems protective. Anything driving risk?
Conversely, fatty acid-binding protein 4, FABP4, which is involved in fat metabolism and inflammation, was found through MR to elevate gastric cancer risk, or R, around 1.21. These findings point towards specific inflammatory pathways and molecules that appear to either protect against or actively promote gastric cancer development, offering potential targets.
And since we touched on the gut microbiota earlier influencing metabolites, are there any more direct causal links emerging from MRR studies, specifically looking at bacterial genera and gastric cancer? How does our gut health seem to influence GC?
Yes, studies are increasingly applying MR to explore the gut microbiome's potential causal role, although it's complex because defining genetic proxies for specific bacteria is tricky.
Yeah.
While some findings can be conflicting or need more replication, for instance, one study might find a link for Ruminocaceae UCG002, and another might not. Some potentially causal links have been identified. For example, the genus Egerthella was found in one MER study to be negatively associated with gastric cancer risk, or quite low, 0.46, suggesting a possible protective effect.
Interesting. So manipulating Egerthella levels could be beneficial.
That's the kind of long-term implication, yes. This sort of discovery could potentially open doors for novel microbiota-modifying therapeutics, maybe prebiotics or probiotics aimed at boosting protective bacteria like Egerthella to reduce cancer risk. Still early days, but promising.
That's exciting for potential future interventions using our own microbes. What about pre-existing conditions? Does having certain autoimmune diseases, for instance, causally impact gastric cancer risk, according to Ms.
Yes, the research points to this, particularly in certain populations. In East Asian populations, specifically, ulcerative colitis, UC, which is a chronic inflammatory bowel disease, an autoimmune condition showed a significant causal association with gastric cancer risk, or around 1.124.
So having UC increases your risk of gastric cancer, causally.
That's what the MR evidence suggests for East Asians. It indicates a heightened risk for individuals with this autoimmune condition. It might be due to shared inflammatory pathways, general immune dysregulation, or maybe even defective treatments for UC. But it points to a causal link between these seemingly disparate inflammatory diseases.
That's an important clinical connection to be aware of. And finally, just throwing this out there, on the more unexpected side, I'm curious if there's any link to hormones, like testosterone.
Interestingly, yes, there was a finding related to that.
Yeah.
Genetically predicted total testosterone levels were found to be a suggestive protective factor against stomach cancer, specifically in men.
Suggestive, not definitive.
Right. The odds ratio was 0.66, which is quite protective, but the statistical significance was borderline, p=0.01116, so it didn't quite meet the strictest criteria some researchers use. However, it's an intriguing signal.
So higher testosterone might protect men from stomach cancer.
It's a possibility suggested by the genetics, yes. It definitely warrants deeper investigation into the hormonal landscape related to gastric cancer risk and potential differences between sexes.
Suggestive indeed. It just goes to show how many different biological systems, metabolism, inflammation, immunity, hormones, might be subtly influencing cancer risk. Any other broad biological pathways popping up in these MR or other large-scale genetic studies that point to fundamental cellular processes involved?
Yes, a couple more worth mentioning. The autophagy lysosome pathway came up. This is a fundamental cellular process involved in basically cellular cleanup and recycling, getting rid of damaged components and maintaining cellular health.
Essential housekeeping.
Very essential. And the inheritance of genetic variants within this overall pathway showed a significant association with gastric cancer susceptibility in one analysis. This highlights that disruptions in even these fundamental cellular maintenance processes, influenced by our genetics, can contribute to cancer risk.
So if the cleanup crew isn't working right,
Things can go wrong, potentially leading to cancer initiation or progression. Additionally, matrix metalloproteinases, MMPs, have been implicated.
NMPs, aren't they involved in breaking down tissues?
Exactly. They're enzymes involved in remodeling the extracellular matrix that scaffold around cells. They play roles in normal processes, but are often hijacked by cancer cells to enable invasion and metastasis. A meta-analysis looking at genetic variations, polymorphisms, in MMP genes found that a specific variant in MMP7, the NEGUS181AG variant, had a significant association with gastric cancer susceptibility, reinforcing their potential role in the progression of digestive system cancers. 4. Evolution of understanding, a timeline of discovery.
It's incredibly clear that the field of gastric cancer genetics is just so dynamic it's constantly evolving. We've talked about a lot of cutting-edge research today, but let's just take a step back for a moment and appreciate the journey itself. How has our understanding of gastric cancer genetics actually evolved over time? From those very early foundational insights to where we are today with MR and PRS, what's the narrative of this scientific discovery been like?
It's been a fascinating progression, really. The journey into gastric cancer genetics truly began in earnest with the advent of those pioneering genome-wide association studies, GWAS, which laid the crucial groundwork back in the late 2000s and early 2010s.
So about 15 years ago.
Roughly, yes. A foundational example often cited is a significant review from 2010 that discussed early genome-wide analyses and the GMDBJ database, using gastric cancer as a prime example. These early studies were absolutely instrumental in identifying the very first convincing susceptibility loci regions in the genome linked to risk. Notably, genes like MUC1 and PSCA were among the first hits.
Okay, so MUC1 and PSCA were among the earliest flags raised in the vast genetic landscape, really putting gastric cancer on the map for genetic researchers and pointing them in specific directions.
Exactly. They provided the first solid footholds.
What happened next? Did the scope of discovery broaden quickly, or did scientists just dig deeper into those initial genes for a while?
Oh, the scope absolutely broadened, and the pace of discovery really accelerated significantly after those initial findings. We moved beyond just confirming those first few associations to identifying a rapidly growing number of new risk loci.
More hotspots emerged.
Many more. This includes those major players we've explored today, like the regions near PRKA-ON on chromosome 5 and UNC5CL on chromosome 6. And then more recently, Truly novel genes identified through GWAS and follow-up studies, such as DOGLB, BTN3A2, FBECO43, and that long non-coding RNA INC00240 we discussed. Each new discovery adds another intricate piece to this complex puzzle, revealing more about the genetic architecture of the disease and showing us entirely new biological avenues to explore.
And as we found more and more of these loci, I imagine that the understanding of the disease itself must have become more nuanced, almost like identifying different flavors or subtypes of gastric cancer based on genetics. Is that where subtype-specific precision really came into its own?
Precisely. That's been a critical evolution in thinking. There's been an increasing recognition of this genetic heterogeneity, realizing that gastric cancer isn't just one monolithic disease genetically. This understanding is based both on the histological subtypes, how the cancer cells look under a microscope,
Right.
like intestinal versus diffuse,
Right.
and also based on the anatomical location where the cancer starts cardia versus non-cardia. This growing precision, driven by larger GWAs designed to look for these differences, has allowed for the identification of loci, like the one in C12 or 51, affecting survival only in intestinal type, that shows specific associations for particular subtypes. This enhanced granularity is absolutely crucial for developing more targeted research questions and looking ahead more personalized clinical approaches. We're moving away from just gastric cancer to a much more granular understanding.
And it's not just the DNA sequence itself that's important, as you've emphasized, but also how it's expressed and regulated. That brings us back to epigenetics. When did that layer become a significant part of our understanding of gastric cancer genetics? Was that always considered, or is it newer?
It's become much more prominent in recent years. The field has made significant strides in incorporating epigenetic modifications, particularly DNA methylation, into our broader understanding. While the concept of epigenetics isn't new, applying it systematically to large gastric cancer cohorts is relatively recent. And this has revealed how these dynamic factors, like the hypermethylation, the silencing of the PRKCB gene promoter, or that specific CpG site, CG1607185, potentially linked to PCB exposure. How these changes can profoundly influence gene expression levels and, consequently, impact gastric cancer risk and prognosis. This adds a crucial layer of regulatory complexity, showing that gene activity can be significantly altered without any changes to the underlying DNA sequence itself. This area is rapidly revealing potential new diagnostic markers and maybe even therapeutic targets aimed at reversing epigenetic changes.
So the story isn't just about the genes you inherit, but how those genes are being actively read or silenced by these chemical marks, and critically, that these marks can even be influenced by our environment over time. That's a huge shift in perspective from just static DNA sequence.
It absolutely is. It introduces a dynamic element influenced by time and exposure.
And then we have Mendelian randomization, which you've highlighted several times as a remarkably powerful tool for trying to get at causality. When did that really rise to prominence in this field? Was there a point where it suddenly allowed for those aha moments of potential cause and effect?
The widespread application of Mendelian randomization, MR studies, to gastric cancer has been a significant methodological shift, especially gaining momentum in, say, the last 5 to 10 years. While the underlying concept has been around for longer, its robust application really took off as the necessary large-scale GWAS data sets for both exposures, like obesity, metabolites, inflammation markers, and gastric cancer outcomes became publicly available.
So the data availability was key.
Absolutely. This powerful approach, using genetic variants as those natural instruments or proxies, allows researchers to establish potentially causal links between various exposures and GC risk. As we discussed, MR studies effectively mitigate many of the traditional confounding and reverse causality issues that plague purely observational studies, providing much more robust evidence. This has been instrumental in clarifying, for example, the likely causal role of obesity, or that surprising potential protective effect of total cholesterol, which observational studies might struggle to pin down definitively.
That's where we really try to move from just seeing a correlation to understanding true cause and effect, which is transformative for prevention strategies. And finally, how has all this accumulating genetic knowledge from GWAS to epigenetics to MIR translated into more practical applications, like actually predicting risk for individuals? How did PRS evolve out of this?
The development of polygenic risk scores, PRS, represents perhaps the most direct translational leap forward from all this accumulated GWAS data, aiming for personalized risk prediction. By integrating the cumulative effect of many genetic variants identified in GWAS, PRS can now identify individuals at significantly higher or lower inherited genetic risk for gastric cancer.
Summing up all those small effects.
Precisely. But what's truly transformative, and perhaps the most empowering message coming out of recent PRS research, is that compelling demonstration we talked about. Combining high genetic risk with adherence to a favorable lifestyle can significantly reduce the absolute risk of developing the disease.
Right. Genetics isn't destiny.
Exactly.
Yeah.
This revelation opens entirely new doors for tailored prevention strategies. We're moving beyond general public health advice, eat well, don't smoke, towards a future of potentially more precision public health, where we can identify high-risk individuals and offer them more targeted advice and interventions, offering tangible hope and clear actions they can take.
So it's not just about knowing your genetic risk score, but knowing that you can potentially do something about it, which is incredibly empowering for people.
That's the key message. Overall, the evolution of understanding in gastric cancer genetics has moved towards a much more holistic and integrated view. It profoundly emphasizes this intricate interplay between our inherited genetic predispositions, critical environmental factors like H. pylori infection and diet, our daily lifestyle choices, and even these dynamic epigenetic modifications. This multi-layered interactive perspective offers a much more complete picture of why gastric cancer develops and, importantly, illuminates promising new avenues for personalized intervention and prevention. It's truly an exciting and rapidly moving time in the field.
The big picture and future directions.
Okay, we've covered a remarkable amount of ground today, really peeling back the layers of gastric cancer genetics in this deep dive. Let's try and bring it all together now, synthesize the key points. If you had to give us a snapshot, like the core takeaways from this whole exploration into the genetic landscape of gastric cancer, what would they be?
Right, boiling it down. I think the first core takeaway is the consistent identification of several critical genetic hotspots. We've seen these recurrent regions pop up again and again in different studies, like chromosome 5p13.1, which involves the PRKAA1 gene, and chromosome 6p21.1 near UNC5CL.
They're frequent flyers.
Exactly. Beyond these specific loci, established genes like MUC1 and PSCA have been repeatedly implicated. And then there are the newer players we discussed showing strong evidence, such as the non-coding RNA LeanNC00240 and genes like DAGLB, BTM3A2, FBXO43, C1GLT1, and the epigenetically regulated PRKCB. These represent crucial nodes in our current understanding of genetic susceptibility.
So there's a pretty consistent genetic map emerging, highlighting key areas in the human genome that influence risk. What's the next big takeaway from all this research?
Second, building on what we just discussed, it's crystal clear that the genetic landscape of gastric cancer is not uniform. It exhibits significant subtype specificity. We see different genetic variants, or maybe the same variants having different effects, depending on the specific histological subtype, like intestinal versus diffuse, and also depending on the anatomical location within the stomach cardia versus non-cardia.
So precision matters.
Absolutely. This level of precision is crucial for driving more targeted research questions, and ultimately for developing more effective, personalized clinical approaches. We can't treat all gastric cancers as genetically identical.
And as we saw with the comparisons between different ancestries, genetics is also influenced by our background. Discoveries in one group might not directly apply or apply in the same way to another.
That's right. The third key takeaway is that there are significant population nuances. While some genetic associations, maybe for fundamental pathways, are broadly shared across humanity, there are distinct differences, particularly when comparing East Asian and European ancestries, but also within those groups. Factors like the prevalence and specific virulence of local H. pylori strains interacting with distinct population-specific genetic backgrounds likely contribute to the differing incidence rates and maybe even different disease characteristics observed globally. Understanding these differences is absolutely key for developing truly global health strategies and interpreting research findings correctly.
Right, context is everything.
Yeah.
And the tools we now have to make sense of all this incredibly complex data, GWAS, METCHO, R, PRS, they're truly revolutionary in how they're changing our capabilities, aren't they?
Indeed. Fourth, the power of prediction is growing exponentially, largely thanks to tools like polygenic risk scores or PRS. These are rapidly emerging as powerful means to stratify individuals based on their overall inherited genetic risk. They offer a tangible glimpse into a future of truly personalized prevention strategies that were honestly unimaginable just a decade or two ago. The ability to identify those at highest inherited risk is potentially a game changer for screening and prevention.
And finally, maybe the most important shift in thinking, it's not just about identifying a genetic variant itself anymore, but really understanding what that variant does at a deep biological level.
Exactly. The fifth point is that the field is increasingly focusing beyond just identifying SNPs towards understanding their full functional implications. This means digging into their impact on gene expression levels, like those EQTLs, how they might influence DNA methylation patterns or other epigenetic marks, and how they ultimately affect protein function or pathway activity. This deeper dive into the precise biological mechanisms is absolutely essential for translating simple statistical associations found in GWAS into actionable clinical insights and, hopefully, new therapeutic targets. We need to know why the association exists.
That's a fantastic snapshot of the core takeaways. Very clear. Now let's get into the specifics again, but maybe in a more synthesized way. Based on everything we've covered today, what are the top genes that keep coming up? The most consistently implicated pathways, and what are the core biological hypotheses that seem to emerge from this entire body of research when you put it all together?
Okay, focusing on the most recurrent and seemingly impactful genes, we definitely have MEC1, PSCA, PRAA1, UNC5CL, NKRD50, and PTGR4 frequently cited in major GWAS. Among the emerging genes that are gaining significant attention and showing strong functional links are DAGLB, BTM3A2, FBXO43, the non-coding RNA linin C00240, C1GBAL-T1, the epigenetically regulated PRKCB, and potentially TMX1. These genes represent critical areas of ongoing investigation and potential future therapeutic focus.
Okay, quite a list there. And which biological pathways are consistently popping up as central to gastric cancer development, almost like the main highways of disease progression that these genes plug into?
Several key pathways consistently stand out across multiple studies and approaches. First, as you might expect, inflammation and immune response pathways are almost universally implicated. This involves components like IL-6 signaling. We saw IL-6R having a protective MR signal, IL-17 signaling, like the IL-17A variant, and maybe the fast signaling pathway involved in cell death. The finding for fatty acid-binding protein 4 also fits here. It profoundly reflects cancer's inflammatory origins, especially with H. pylori.
Makes sense. What else?
Second, cell adhesion and proliferation pathways are absolutely central. Genes like MUC1 are involved here, and generally processes regulating how cells stick together, or don't, allowing invasion, and pathways controlling uncontrolled cell growth, where TMX1 might play a protective role, are fundamental to tumor development and spread.
Okay, growth and sticking.
Third, epigenetic regulation, as we've discussed, particularly DNA hypermethylation silencing genes like PRKCB or specific sites like CG1600 ESRI7185, represents a critical dynamic mechanism impacting gene expression and risk. It's a key regulatory layer. Fourth, metabolic reprogramming is clearly important. This is highlighted by the involvement of specific metabolites identified through MR, like 3-methyl-2-oxyvalerate, and those causal links identified for obesity, risk factor, and total cholesterol, potential protective factor. It points to significant metabolic contributions to GC development.
So how the cell uses energy and building blocks.
Exactly. Fifth, various crucial intracellular signaling cascades are consistently enriched in pathway analyses of GWAS's hits. These include the usual suspects like the p53 tumor suppressor pathway, MAPK signaling, and TGF-beta signaling, along with others like PI3K-AKT, often linked to growth, and general cytokine signaling, often linked to obesity and inflammation. These underscore the complex intracellular communication networks driving gastric carcinogenesis.
A lot of crossed wires in there.
Definitely. And finally, let's not forget the autophagy lysosome pathway. The finding that genetic variants in this fundamental cellular recycling and cleanup process contribute to GC susceptibility highlights how disruptions in even basic cellular maintenance can have dire consequences.
That paints a really comprehensive and, frankly, quite intricate picture of the underlying biology. So bringing it all together now, what's the overarching biological hypothesis or framework for how gastric cancer develops? Based on all these cutting-edge genetic and functional findings, how does it all seem to connect?
Okay, the core biological hypothesis, as it stands now, is that gastric cancer develops through a complex and highly interactive interplay of inherited genetic variants the hand you're dealt. These variants fundamentally affect key cellular processes. Things like cell proliferation, how our immune system is regulated in response to threats, background inflammatory responses, and crucial metabolic pathways.
So the genetic foundation.
Right. But crucially, and this is key, these inherited genetic predispositions are almost always significantly modulated by environmental factors. Most notably, Chronic H. pylori infection is a massive player, but also an individual's diet, smoking habits, alcohol intake, obesity, and broader lifestyle choices are critical modulators.
Genes plus environment.
Exactly. And layered on top of that, as we've learned, epigenetic modifications play a crucial dynamic role in regulating gene expression in response to some of these environmental cues or just stochastic events, thereby influencing both cancer risk and patient prognosis. So it's truly a multi-layered interactive process where our inherited genes constantly dance with our environment and our lifestyle choices over time.
That's a fantastic summary. Very clear. To wrap up this synthesis section, can you just give us some final comparative notes? Remind us of the key distinctions we need to keep in mind across different populations and those histological subtypes as we think about the global picture of gastric cancer.
Certainly, it's crucial to remember that while some major gastric cancer susceptibility loci, perhaps those involved in very fundamental pathways like near PRKAA1 or MUC1, show broad associations across diverse populations, their specific impact, the size of the effect, the frequency of the risk allele, or even the precise statistical model of association, like dominant versus recessive for IL-17A, can indeed differ significantly across populations.
So the details matter.
Very much so. For instance, East Asian populations clearly have some specific genetic associations identified, like that IL-17A variant, behaving differently. The H. pylori interacting SNPRS 2671655 being prominent there, and the C12F51 variant linked to intestinal subtype survival. These unique genetic predispositions, possibly interacting with local environmental factors like specific H. pylori strains, may contribute to the higher incidence rates and perhaps distinct disease characteristics often observed in East Asian populations compared to, say, Europeans.
And H. pylori factor itself varies too, right? The bug isn't the same everywhere.
Exactly. The importance of specific H. pylori virulence factors, like the completeness of that CAG-PI pathogenicity island we talked about in Mongolia, clearly varies by geographic region. And critically, these bacterial factors show unique interactions with the host's genetics. This emphasizes again that it's not just the presence of the bacteria, but its specific genetic makeup and how it interacts with the host's own DNA that likely determines the ultimate risk level.
Okay. And the subtypes within the cancer itself.
Lastly, yes, those clear, distinct genetic contributions we're seeing for cardia versus non-cardia gastric cancer and for intestinal versus diffuse types fundamentally emphasize the critical need for precision medicine approaches. We need to move beyond treating gastric cancer as a single entity. Understanding these nuanced genetic differences allows for potentially more tailored prevention advice, more accurate diagnosis perhaps using molecular subtyping, and ultimately the development of more effective and personalized treatment strategies. It really pushes us away from a generalized approach towards one that acknowledges the unique genetic and molecular blueprint of each tumor and each patient. Outro.
Wow. What an incredible journey we've taken together today. We've truly gone on a deep dive, haven't we, into the fascinating, sometimes bewildering, but ultimately crucial world of gastric cancer genetics.
It's certainly a lot to take in.
It is. We've moved from understanding these broad genetic associations found in huge populations to pinpointing specific genes, mapping out complex biological pathways, and really starting to unravel the powerful, dynamic interplay between our very own DNA and the world around us, our environment, our infections, our lifestyle.
Yeah, we've seen how those massive genome-wide association studies, the powerful Mendelian randomization analyses trying to get at cause and effect, and all the detailed functional lab work, how they're all contributing to painting an increasingly detailed picture of who is at risk for this disease and, importantly, why.
It's been a profound exploration, I agree. Moving from the microscopic level of the genetic code right up to the macro level of population health and lifestyle choices. The depth and breadth of the current research really is remarkable, bringing us closer than ever to understanding a disease that has historically been so challenging and often deadly.
It offers hope, definitely.
So what does all this complex science mean for you, the listener? Well, the ability to potentially identify high-risk individuals through these cutting-edge tools like polygenic risk scores, combined with a much clearer understanding of crucial gene-environment interactions, especially that critical one with H. pylori infection. This isn't just academic anymore.
No, it has real-world implications.
Exactly.
Yeah.
It means we're moving closer, step by significant step, towards developing truly personalized prevention strategies for gastric cancer. Imagine knowing your risk profile and getting tailored advice based on it.
And perhaps even more importantly, that revelation we discussed that positive lifestyle choices can significantly reduce risk even in those individuals with a high genetic predisposition. That's perhaps one of the most powerful and actionable insights from all this research.
It really is. It empowers individuals, doesn't it?
Yeah.
To take proactive steps to make changes regardless of the genetic hand they've been dealt. It offers tangible hope for prevention through action.
Absolutely. Your genes load the gun, as they say, but environment and lifestyle often pull the trigger, and you have some control over the latter.
Absolutely. It's a profound call to action for anyone looking to optimize their health and potentially reduce their cancer risk. And that brings us quite nicely to our final provocative thought for today. If our genetic blueprint, combined with understanding these interactions, can tell us so much about our susceptibility to complex diseases like gastric cancer, what other hidden biological messages are just waiting there to be uncovered in the vast, vast ocean of our own genome? What more can we learn that could truly guide us toward healthier, more resilient lives through actionable, personalized insights derived directly from our own biology? This deep dive today feels like just the beginning of that ongoing discovery, doesn't it?
An incredibly exciting future awaits as we continue to unlock the secrets held within our own DNA and learn how to use that knowledge wisely.
Couldn't agree more. Thank you so much for joining us on this deep dive. We sincerely hope you feel incredibly well-informed, maybe a little amazed, and perhaps inspired by the rapid advancements happening in this vital field of research. Until next time.